Hi, let us in this video try to understand some of the homeostatic regulatory mechanisms. Basically, all the organ systems have only one goal, and that's achieving and maintaining the homeostasis. And therefore, it's very important for us to understand how these homeostatic regulatory mechanisms work and how organ systems are functioning and striving to achieve that homeostasis. Before that, let's just define two things. First, myelo-interior. Myelo-interior is the internal environment of the body, and internal environment of the body is same as extracellular fluid, because the cells are bathed in extracellular fluid, cells are surrounded by the extracellular fluid, and therefore that is the myelo for the cells. This term was coined by Claude Bernard, and this therefore is the myelo for the cells. Now, second definition, the homeostasis. This term is attributed to another well-known physiologist, Walter F. Cannon, and this means maintaining the constancy or stability of the myelo-interior. The myelo needs to be kept stable and constant, because then the cells will function optimally. So, maintaining the constancy or stability in the myelo-interior is homeostasis. And all organ systems, they try and maintain this constancy or stability. How does this homeostasis work, or how it is maintained, the stability or constancy? It's based on the regulatory mechanisms. Before we actually talk about the regulatory mechanisms, let us understand the schematic model of those regulatory mechanisms. In the ECF, there are controlled variables. In the myelo, there are controlled variables, the parameters. For instance, pH is one controlled variable, temperature is another controlled variable, osmolarity, the glucose levels, O2 levels, CO2 levels, all these are controlled variables in the ECF. And for each of them, there is a sensor. The sensor can sense the value of the controlled variable in the ECF. In the myelo, that value is constantly being sensed, and accordingly the signals are sent to the controller. Let's take an example. For instance, let's say blood pressure is a controlled variable. The sensor is baroreceptor and it sends the signal to the controller. The constant value is being sensed, and any disturbance in the myelo, any disturbance and change in the control variable is also sensed and sent to the controller. The controller already has the set point for that control variable. It knows the set point. So, controller will compare the value of set point and this disturbance which has happened now, and accordingly the controller will take the action via the effector organs. In the case of blood pressure, we said baroreceptors are the sensors, vasomotor center is the controller, then effector organs will be heart and blood vessels. So that's the schematic model, and based on this, all the organ systems and regulatory mechanisms function. The regulatory mechanisms are of two types. At a very basic level, two types. One is called as feedback mechanism, feedback regulation, and the other one is called as feed forward regulation. Feedback mechanism is the one in which the change occurs in the control variable. The change occurs in the milieu, and that change is fed back to the controller. The change is fed back to the controller, and then the controller takes the action. This will be the feedback type of regulatory mechanism. Change occurs, remember this, that change is fed back and then the controller will act. Whereas feed forward means the change is anticipated. Change does not happen, change is anticipated, and even before that change has actually happened, the action is taken. So the controller takes the action anticipating a change. The change, before the change could happen, the controller actually takes the action. That's a feed forward regulation. Let's understand the examples. Feed forward, common example is cephalic phase of the gastric acid secretion. Common example. Just by sight, smell or thought of the food, there is gastric acid secretion is initiated. That's a feed forward. I mean, this is anticipated now that the food will be taken and food will go into the stomach, and then the gastric acid will be anyways needed. So in anticipation of that, the feed forward, the controller has already acted. So that's feed forward. Whereas feedback, most of our mechanisms are feedback. For example, temperature increases, then it is brought back to normal by sweating or cutaneous vasodilation. That's a feedback. Change occurred, and then the change was then it was fed back, and then the controller took the action. Let's see those mechanisms one by one. Feedback as well as feed forward are again of two types, each of them. Feedback is negative feedback and positive feedback. Feed forward is also negative feed forward and positive feed forward. Let's understand these one by one. Feed forward. Positive feed forward we can explain very easily. When we say positive or negative feed forward, the meaning is either the effect which is happening in anticipation of the event, that effect will be either stimulatory, excitatory, when we will call it positive feed forward, or it is inhibitory, it will inhibit in anticipation, then that will be called as negative feed forward. So examples of positive feed forward, we saw one example just now, cephalic phase of the gastric acid secretion. Even before the food has gone into the stomach, gastric acid secretion starts just by thought or sight of the food. That's a positive feed forward. Or increased ventilatory drive just at the start of exercise. This is positive feed forward. When we perform the muscular exercise for, let's say, about 3-4 minutes, and after that the ventilatory drive increases, that's understandable because we are performing the exercise and the muscles are generating the CO2. Now this increased CO2 levels, the CO2 will cross the blood-brain barrier, it will stimulate the respiratory center, the ventilatory drive will increase, and the CO2 will be washed out of the body. This is a negative feedback. It's a feedback and it's a negative feedback. CO2 is washed out. So this is understandable. How come just at the start of exercise the ventilatory drive increases? That's a feed forward, positive feed forward. That is, the controller anticipates that the exercise is starting, so the muscles will need extra oxygen. and therefore the controller immediately starts its action and ventilatory drive will be increased. Cortex is sending the signals to the muscle to start the contraction and start the exercise. There will be collaterals sent to the brainstem, respiratory center, and ventilatory drive will be increased just at the start of exercise. So this is a feed forward regulation in anticipation of the change. Or one more example is action of parathyroid hormone via vitamin D to increase the serum calcium. You know, we have the calcium sensors in our body. They are sensing the moment to moment changes in the calcium levels, or they are sensing the calcium levels very regularly, very frequently, and just at the anticipation, almost like an anticipation that the calcium is going to fall, the serum calcium is going to decrease, the parathyroid will come into action. It will remove the calcium from the bone and will be brought into the circulation in the serum calcium to increase, but also it will stimulate the secretion of vitamin D, vitamin D synthesis rather. Vitamin D action will be potentiated, and that will increase the calcium absorption rather than increasing the synthesis or secretion. Actually what happens is parathyroid hormone potentiates the action of vitamin D of increasing the calcium absorption from the gut. So this is a positive feed forward. It's almost like an anticipation that the calcium is going to fall and it will go to a threatening level, life-threatening level. So parathyroid acts in a very rapid manner, in a positive feed forward manner. Negative feed forward or feed forward inhibition. Feed forward inhibition is seen in cerebellar circuits in the context of motor learning. Cerebellum is the master coordinator for our voluntary movements, and it has circuits, and those circuits are so finely balanced that we perform the voluntary movements in a very precise, smooth and well coordinated manner. One type of circuit in the cerebellum is of feed forward inhibition. There is a Purkinje cell in the cerebellum, and Purkinje cell exerts its inhibitory influence on the deep nuclear cell. Deep nucleus sends output from the cerebellum. Now then there is a granule cell in the cerebellum. This granule cell, after receiving afferents coming into the cerebellum via mossy fiber, then granule cell gives short axons which run to the surface layer and they form a T. These are called as parallel fibers. Granule cell excites the Purkinje cell. Now if the Purkinje cell were to inhibit the granule cell, we would call it feedback inhibition. But granule cell, even before it could excite the Purkinje cell, via short inhibitory interneurons like Golgi cell, basket cell or stellate cell, Golgi cell, stellate cell, basket cell, inhibitory interneurons, granule cell will excite those interneurons and they in turn will inhibit the Purkinje cell. So granule cell, before it could excite the Purkinje cell and then that having the effect on the cerebellar output, even before that could happen, granule cell can inhibit the Purkinje cell via these interneurons. This has been termed as feed forward inhibition. What is the importance of this? Because of such finely balanced circuitry, cerebellum can precisely decide which muscle to contract how much, and this is happening during learning process. When we are learning after birth to make the voluntary movements slowly, we make the errors and we learn that how much to contract an agonist, how much to antagonist should contract or relax so that the movements can be precise smooth and very well coordinated. There is no jerkiness in the movement. We don't overshoot the target. We don't stop short of the target because of this finely balanced circuit in the cerebellum. So this is feed forward inhibition. Now coming to the feedback mechanisms. Feedback mechanisms, as we have already mentioned, these mechanisms work in the way that the change occurs in the control variable. That change is fed back to the controller and then the controller will take the action. So change definitely happens and then the controller is going to act. That's the feedback type of mechanism, regulatory mechanism. Blood pressure, for example, it's changed and then the action is taken and it is brought back to normal. It's of two types: negative feedback and positive feedback. Let's understand them one by one. First, the negative feedback. Most of the body's homeostasis, 99% of the homeostatic regulatory mechanisms, they work on a negative feedback principle. That the change occurs and that change is negated, cancelled. So what happens is, controller, when it receives the change, controller acts in the opposite direction to that of the change. If the change has occurred in a certain direction, controller will act in the opposite manner so that the change is nullified, the change is negated, the change is cancelled out, and the parameter, the control variable, is brought back to the original set point. That's negative feedback. Controller does the opposite to the change which occurred. So BP increased, the controller brings it back to normal or cancels the change. Now because of this, the error is minimized. The change that we have been saying has caused a certain error in the value of control variable. So in the negative feedback, the error will be minimized. Most of the examples in our body that are involved in homeostasis, they belong to this. Osmolarity increased. If the ECF has become hypertonic, we will feel thirsty. We will increase the fluid intake, the water goes into the plasma and the tonicity will be normalized. So hypertonic plasma became normotonic again. So basically every other mechanism is of negative feedback. The other type is positive feedback. Before we move on, let me just say, we will discuss this error being minimized or amplified and efficiency, how efficiently it is done. We will discuss that in the next video. The efficiency of homeostatic regulatory mechanisms and efficiency of the negative feedback mechanisms in terms of gain of the controlled system will be discussed in the next video. So positive feedback means controller does the same. Controller acts in the same direction as that of the change. If the change has happened, controller acts in the same direction. So more and more of the same will keep on happening. Change is not negated, change is amplified. So in this, the error is magnified, error is amplified. That is the negative, I beg your pardon, that is the positive feedback. Error gets amplified. It's A leading to B and B leading to A again and A leading to B in a cyclical fashion. That's positive feedback. Obviously, this would not be good for the body because this will increase the deviation from the set point. So, it's sometimes called as vicious cycle. It's vicious, it's not so good to the body. It's vicious cycle. For instance, second stage of circulatory shock. Circulatory shock means let's say because of the hemorrhage or fluid loss, the BP and the cardiac output have decreased and there is inadequate blood perfusion to the tissues. In the first stage, there is negative feedback. The change is happening, BP is falling, and baroreceptors and other mechanisms are trying to cancel it out and bring it to the normal level. But when the bleeding continues and more than 10% blood volume is lost, then the patient goes into second stage of circulatory shock where positive feedback ensues. So what actually happens is there is a decreased blood pressure and a decreased cardiac output. Therefore, there is decreased perfusion in the coronaries, decreased blood flow in the coronaries, so decreased perfusion of the myocardium. Because of that, the myocardium is depressed. Its functioning goes on decreasing. And as the myocardium is depressed, there is a further decrease in the cardiac output. There is a further decrease in the blood pressure. So this goes on happening in a vicious cycle manner. So obviously this would not be deemed to be good for the body. But there are certain examples of positive feedback which are helpful to the body. Let's understand the helpful examples of positive feedback. For instance, oxytocin and its role in parturition. Positive feedback but helpful to the body. At the time of delivery, baby's head starts pressing on the cervix. Signals are sent to the posterior pituitary. There is a release of oxytocin. Oxytocin comes to the uterus, causes contraction of the uterus. Baby is further pressed down and there is further signaling sent to the posterior pituitary, further release of oxytocin. More and more of the same keeps on happening in a cyclical pattern. Or platelet plug formation and clot formation. Again, a positive feedback. That when the vessel is ruptured, initially few platelets come into that area, they are activated, they release certain chemicals, so more platelets will come into that area and they will adhere to the previous ones. They will release certain chemicals, so some more platelets will come into that area. More and more of the same keeps on happening. Or even clot formation. One clotting factor is activated, it will act enzymatically on the next factor is activated, and that will act on the third factor. Positive feedback. Third, LH surge leading to ovulation. First few days of the menstrual cycle, estrogen keeps a negative feedback on the LH and LH levels are low. But just a few days before the ovulation, it enters the positive feedback. That is, estrogen stimulates the LH secretion, LH on the estrogen, and they will act in a positive feedback manner, leading to the LH surge and finally the ovulation. That's a positive feedback. Or bladder filling and micturition. The process of micturition starts as a weak signal or weak process, but it increases in strength. Why? Because it's a positive feedback. It's a self-regenerative process. So it happens like this. First few drops entering the posterior urethra, dilation of the urethral wall, stimulation of the stretch receptors in the wall, that sends the signal to the spinal cord. The signals come back, there is bladder contraction, the urine starts flowing, and the signal becomes stronger and stronger. More and more of the same keeps on happening. So that's a micturition process, starting as a weak process but gradually becoming stronger and stronger and stronger, self-regenerative process. Or sodium channel opening during action potential, a positive feedback cycle. When we apply the stimulus to the nerve, initially a few sodium channels will open. Therefore, some sodium will enter the nerve at that point. Some sodium will enter means positive charges are entering, therefore membrane potential will drop to a less negative value. Membrane potential drops from, let's say, -70 to -68, and that opens some more sodium channels. So some more sodium will enter. Membrane potential will drop further from -68 to -65, and this will go on in a cyclical fashion. Positive feedback. One more, Head's paradoxical reflex. Now we have heard of Herring-Brewer reflexes. In the case of respiratory system, there is a classic negative feedback reflex called as Herring-Brewer reflex. Over distension of the lungs leads to deflation of the lungs. This is to prevent the damage to the lung parenchyma. The Herring-Brewer reflex, a classic negative feedback. But there is also this Head's paradoxical reflex. What does it exhibit? Distension of the lungs leads to more distension. That will lead to further distension, and that will lead to more and more distension of the lungs. This is called as Head's paradoxical reflex. Now this is said to occur at birth. At birth, the lungs are collapsed. They are going to distend after that. With each breath, they are going to distend and distend and distend. And therefore, for that to happen, this positive feedback happens. With each breath, they will be opening more and more and more till they are completely opened up and start functioning normally. So that's a Head's paradoxical reflex. These are all the examples of positive feedback. Last two interesting points that I would like to mention here. One is all the positive feedbacks, they end with negative feedback. Obviously, that has to happen because it cannot go on forever. I mean, take the example of clot. It can, or platelet plug. It cannot go on happening forever. So it has to end somewhere, and therefore that happens by negative feedback. The process of positive feedback will be stopped. It will be curtailed and it will be completely stopped. That's a negative. So every positive feedback toward the end will have a negative feedback process to curtail the positive feedback. And the second, even more interesting point is that all these positive feedbacks should be actually seen as a part of larger negative feedback process. Yes, let's take the example. Let me explain this. Let's say oxytocin in parturition. Positive feedback. Yes, it is happening in a positive feedback manner. But think of this. Here is a lady, here is a female with a certain body physiology. With pregnancy, that body physiology had got altered, it was changed. With delivery, which is happening with the help of this oxytocin and positive feedback, the body physiology will return to normal. The change will be negated, cancelled out. So positive feedback is happening, but actually it is allowing restoration of the set point, or negative feedback to happen in a larger context. Or clot formation, platelet plug formation and clot formation. We have a circulating blood volume. Because the blood vessel is ruptured, that blood volume is going to decrease. By platelet plug formation, the bleeding stops so that the blood volume can return to normal. Negative feedback, the change can be negated. Blood volume had decreased because of the bleeding, it can be restored. That's a negative feedback. So platelet plug formation is a positive feedback. It is happening by positive feedback, but it's a part of larger negative feedback process. Restoration of blood volume, negating the change, cancelling out the change, and blood volume to be restored. So that is another interesting feature of this homeostatic regulatory mechanisms.